Welcome to the Deep Dive, the show where we really try to break down complex stuff and, get to the core ideas.
That's the plan.
Today, we're diving into something absolutely fundamental, how your body manages its internal fluids with incredible precision.
It really is amazing when you stop to think about it.
Like, how does it know exactly how much water, how much salt to keep, and why is getting that balance just right so critical for just staying alive?
It's a fantastic question. And for this deep dive, we've been digging into Chapter 40 of Boron and Boulpaep's Medical Physiology, specifically the bit on integration of salt and water balance.
Right, dense stuff.
Oh, definitely. So our mission today is really to unpack it. We'll start, big picture, then drill down into the details.
And connect it to what you might actually see clinically.
Exactly. Make it relevant. See how this basic physiology plays out in medicine.
Okay, so let's lay it out. The body has, two main control systems for this, right? They work together, but they're kind of distinct.
That's a great way to put it. Yeah, two systems. One's focused purely on regulating the volume of your extracellular fluid, the ECF. That's all the fluid bathing your cells, plasma, interstitial fluid, the works.
And the other?
The other is all about controlling the concentration, the osmolality of that same fluid, keeping it from getting too dilute or too concentrated.
And why do we need both? Why separate systems?
Well, they're vital for different things. ECF volume, that's absolutely key for blood pressure, for making sure enough blood actually gets pushed out to your tissues, perfusion. Without that, organs don't get oxygen, nutrients, game over.
Okay, volume equals pressure and flow. Got it.
Then osmolality, that's critical for your cells. If the ECF gets too concentrated, water rushes out of cells and they shrink. Too dilute, water rushes in, they swell.
And I guess that's especially bad for the brain.
Extremely bad. The brain's encased in the skull, there's no room for swelling. Even small shifts in osmolality can mess with nerve function pretty severely.
So different goals, volume versus concentration. But you said they're intertwined.
Very. And here's the cool part. While they use different sensors, different hormones mostly, they share one crucial effector organ, the real MVP.
Let me guess. The kidney.
The kidney, exactly. It's the kidney that ultimately adjusts how much sodium gets excreted to control volume and how much water gets excreted to control osmolality. It's this elegant duet.
Okay, let's tackle the volume part first. Sodium. You called it the master of ECF volume. Why sodium specifically?
Right. So sodium, along with the anions that usually travel with it, like chloride and bicarbonate, is by far the most abundant solute outside your cells.
Okay.
And because water moves freely across most cell membranes to equalize concentration wherever sodium goes, water pretty much has to follow. Water chases salt, essentially, to keep the osmolality stable.
So if you control the total amount of sodium in the body, you're indirectly controlling the volume of water outside the cells.
Precisely. Since the body keeps ECF osmolality in such a tight range, the total amount of sodium must dictate the ECF volume. The kidneys, by controlling how much sodium you pee out versus how much you keep, are basically turning the volume knob for your whole extracellular space.
And those examples from the book really drive this home, like holding on to just 140 millimoles of sodium.
Yeah, which isn't actually a huge amount, chemically speaking.
Right, but it forces the body to retain a whole liter of extra water to keep the concentration the same.
Yeah, boom. You've gained a kilogram, about 2.2 pounds, just like that. Mostly water weight tied to that salt.
Wow. So small sodium changes, big volume effects.
Exactly. It highlights how critical precise kidney control over sodium excretion really is.
Now, you mentioned something called effective circulating volume, ECV. It's not just the total ECF volume the kidneys are sensing.
No, and this is a really key distinction. ECV isn't just the total volume in the tank, so to speak. It's more like the functional pressure and fullness within the blood vessels, particularly the big ones in your chest, the thoracic circulation. It reflects how well the volume you have is actually perfusing tissues.
Okay, so it's about the effectiveness of the volume. Give me an example.
Sure. Think about congestive heart failure. A patient might have massive edema, right? Swollen legs, maybe fluid in the lungs. Their total ECF volume is way up.
Yeah.
But because their heart's weak and not pumping efficiently, the effective circulating volume, the pressure sensed in those key vessels, is actually low.
So the kidneys get the wrong signal.
Exactly. The kidneys think, oh no, we're low on volume. And they start retaining sodium and water, which unfortunately just makes the edema worse. It's a vicious cycle.
That makes sense. What about that water immersion example?
Oh yeah, that's a classic. Stand someone up to their chin in warm water. The water pressure outside pushes blood from their legs and abdomen up into their chest.
So you're increasing the volume in the thorax.
Dramatically.
Yeah.
Even though their total body water hasn't changed, the sensors in the chest say, whoa, overload. And the kidneys respond by dumping sodium like crazy. It shows how sensitive those thoracic sensors are to changes in central blood volume.
Okay, so the body senses this ECV. If it drops, how does it fix it? You said there are multiple pathways.
4 parallel pathways, essentially. It's a belt and suspenders approach. The sensors are varied baroreceptors, sensing pressure in arteries and veins, specialized cells in the kidney itself, even sensors in the brain and liver. They all feed into this response.
Let's walk through them. Pathway number 1.
The big one. The renin-angiotensin-aldosterone system, or RAAS. This is the hormonal cascade. Starts with the liver making a precursor, angiotensinogen.
Okay.
Then when ECV drops, the kidney releases an enzyme called renin. Renin chops angiotensinogen into angiotensin and others.
Still not active yet.
Not really.
Yeah.
Angiotensin then travels, especially through the lungs, where there's lots of an enzyme called ACE, angiotensin converting enzyme.
Ah, ACD inhibitors block that, right?
Exactly. AE converts angiotensin I into the really active form, angiotensin II, or ANGAA2. There's also an ANGAPO, but ANGI2 is the main player here. He has a short half-life, so its effects are potent but controllable.
So what triggers the kidney to release renin in the first place? How does it know ECV is low?
3 main ways. 1, lower overall blood pressure stimulates the sympathetic nerves going to the kidney, telling it to release renin. 2, specialized cells in the kidney tubule, the macula densa, sends less salt flowing past them, signaling low volume. And 3, the kidney has its own internal pressure sensor, a baroreceptor in the afferent arteriole, feeding the filtering unit that detects lower renal blood pressure directly.
So low pressure, low salt delivery, low kidney blood flow, all scream, release renin.
Pretty much. And once you have ANGI2, it goes to work saving salt and water.
Ow. What does it do?
Several things. First, it tells the adrenal glands, sitting on top of the kidneys, to release aldosterone. Aldosterone acts on the kidney tubules to make them reabsorb more sodium.
Okay, so ANGI2, aldosterone, all sodium retention.
Right. Second, ANGA2 is a potent vasoconstrictor. It tightens blood vessels, which raises blood pressure directly. It also specifically tweaks blood flow within the kidney to favor reabsorption, increases the filtration fraction, and reduces blood flow in the medulla.
Clever.
Third, ANGI2 directly stimulates sodium transport, like the NADH exchanger, in the proximal parts of the tubule. And fourth, and this is a key link, it acts on the brain to stimulate thirst and the release of AVP, the water-regulating hormone.
Ah, so it bridges the volume and osmolality systems.
It does. It's a crucial intersection.
And this RAAS system is a big deal clinically, right? Like with hypertension.
Absolutely. Think about renal artery stenosis, narrowing of the artery supplying a kidney. That kidney senses low blood flow, cranks out renin, activates RAAS, and drives up systemic blood pressure. That's why ACE inhibitors, or angiotensin receptor blockers, ARBs, are often first-line treatments for hypertension, especially if RAS is suspected to be overactive.
Okay, that's RAAS, pathway 2.
Increased sympathetic nervous system activity, your fight or flight response. When ECV drops, sympathetic outflow increases.
And that affects the kidney how?
Directly, it stimulates sodium reabsorption in the tubules and constricts renal blood vessels. Indirectly, it also stimulates renin release, giving RAAS another boost. It's especially important in acute situations, like hemorrhage, helping clamp down and conserve volume fast.
Makes sense. Pathway 3.
Arginine vasopressin, AVP, also known as ADH, antidiuretic hormone.
But isn't that mainly for water?
Primarily, yes.
Yeah.
But when ECV takes a big hit, like a major hemorrhage, AVP levels go way up. And at those high levels, it also contributes to vasoconstriction and potentially sodium retention. The body pulls out all the stops when volume is critically low.
Okay. And the fourth pathway?
This one works in reverse, sort of. It involves atrial natriuretic peptide, or ANP. ANP is released by the heart's atria when they get stretched by high blood volume.
So ANP normally makes you lose sodium.
Exactly. It promotes sodium and water excretion, lowers blood pressure, inhibits renin and AVP. It's the "we have too much volume" signal.
So if ECV is low, the atria aren't stretched.
ANP release goes down. And removing that inhibitory signal indirectly promotes sodium retention. So less ANP helps conserve volume.
4 pathways, all working together to bring low ECV back up by retaining sodium. Okay. What happens when things go wrong long-term, like chronic sodium retention?
If the kidneys can't excrete sodium properly, maybe due to kidney disease or severe heart failure, you get chronic expansion of the ECF, plasma volume goes up, but a lot of that extra fluid leaks into the interstitial space.
Leading to edema.
Right, that classic pitting edema you press on the swollen area, usually ankles or lower back, and the indent stays for a bit. Fluid can also build up elsewhere, like pleural effusions around the lungs or ascites in the abdomen.
And the flip side, losing too much sodium.
That can be just as dangerous, if not more so acutely. Things like aggressive diuretic use without proper monitoring or certain kidney diseases that cause salt wasting can shrink the ECF dramatically.
Leading to low blood pressure.
Severely low. Hypovolemic shock. If you don't have enough volume circulating, you can't maintain blood pressure, organs don't get perfused, and things can go downhill very fast. It really shows how vital sodium balance is.
Okay, besides these big four pathways responding to ECV, are there other ways blood pressure itself affects sodium handling?
Yes, definitely. There's a phenomenon called pressure natriuresis or pressure diuresis. Basically, if your arterial blood pressure goes up, even without a change in sensed ECV, the kidneys respond directly by excreting more sodium and water.
How does that work?
It's thought to involve several things. Higher pressure might push more fluid through the filters initially, increasing GFR. It can inhibit RAAS locally, change pressures in the tiny capillaries surrounding the tubules, and maybe directly affect sodium transporters. It acts like a safety valve to help buffer blood pressure rises.
Is there a clinical example of that?
Little syndrome is a good one. It's a rare genetic disorder where kidney tubules reabsorb way too much sodium, causing volume expansion and high blood pressure. Part of what prevents the pressure from going even higher is this pressure natriuresis mechanism kicking in, trying to force out some of that excess sodium due to the hypertension itself.
Fascinating. Okay, we've covered sodium and volume pretty thoroughly. Let's switch gears to the other side of the coin. Water and osmolality.
Right. If sodium is the king of volume, water is definitely the ruler of concentration of osmolality.
And you said the brain is super sensitive to this?
Extremely. Maintaining whole body osmolality within a very narrow range is crucial, largely to protect brain cell volume and function.
So how does the body control water? Two main ways again.
Yep. Kidney water excretion, how much water you pee out in thirst, which drives water intake. Both are controlled by feedback loops starting in the hypothalamus in the brain.
And the main hormone here is AVP.
Arginine vasopressin, AVP, or ADH. That's the primary regulator of renal water excretion.
How was that figured out?
Through some really elegant experiments back in the 1940s by Verney, he showed that injecting a concentrated salt solution directly into the carotid artery going to the brain immediately shut down urine production and antidiuresis.
But injecting it into a regular vein didn't have the same effect.
Not nearly as strong or fast, it proved there were sensors, osmoreceptors, in the brain. Then they found that extracts from the posterior pituitary gland could mimic this effect, leading to the identification of AVP.
And these osmoreceptors are incredibly sensitive.
Amazingly so.
Huh.
If you were to plot plasma osmolality against AVP levels, you'd see AVP stays very low until osmolality hits a threshold, typically around 280 millizomes Gha.
Okay.
Then even a tiny, like 1% increase in osmolality above that causes a sharp, steep rise in AVP release. The system responds very aggressively to small changes in concentration.
What about things like urea? That adds to osmolality, right?
It does, but it doesn't trigger AVP much because it crosses cell membranes easily. It doesn't really pull water out of the osmoreceptor cells, so it's not an effective osmol in this context. It's really about solutes like sodium that are stuck outside the cells and cause water shifts.
So where exactly are these osmoreceptors and where is AVP made and released?
The osmoreceptors are mainly in specialized areas of the hypothalamus, parts called the OVLT and SFO, which are neat because they actually lie outside the normal blood-brain barrier, allowing them to directly sample the blood's concentration.
Ah, clever design.
Very. When these sensors detect increased osmolality, they signal large neurons nearby, the magnocellular neurons, which actually produce AVP. The AVP is then packaged up, travels down long axons to the posterior pituitary, and released into the bloodstream from there.
It acts pretty quickly.
Yeah, it has a half-life of only about 15-20 minutes, so the body can adjust water excretion quite rapidly by turning AVP release up or down.
Okay, AVP handles the output side. What about the input side, thirst?
Thirst is the second key player in osmoregulation. The thirst centers are located in pretty much the same brain areas as the osmoreceptors for AVP, like the OVLT and SFO.
And they respond to the same thing, cell shrinkage from high osmolality.
Exactly. So when osmolality goes up, you get both increased AVP to conserve water and increased thirst to bring more water in. They work together to dilute the body fluids back to normal.
Now, are there things other than osmolality that affect AVP and thirst? You mentioned volume earlier.
Yes, and this is critical clinically. While osmolality is the most sensitive trigger day-to-day, A large drop in effective circulating volume, maybe 5–10% or more, becomes a very powerful stimulus for AVP release.
Even if osmolality is normal or low.
Even then, it can override the osmotic signal.
Hmm.
If you imagine that AVP versus osmolality graph again, severe volume loss shifts the whole curve to the left. AVP starts getting released at lower osmolalities, and the response becomes even steeper.
Why would the body do that? Releasing AVP when osmolality is low seems counterproductive.
Because, as we said before, defending volume often takes priority in extreme situations. In severe hemorrhage or shock, the body floods the system with AVP. Yes, this retains water and might dilute the blood sodium, causing hyponatremia, but the primary goal is to maintain blood pressure and perfusion, even if it means temporarily messing up the osmolality. Volume first, osmolality second in that scenario.
So how does the body sense that big volume drop to trigger AVP?
Through several routes. Stretch receptors in the heart, particularly the left atrium, sense underfilling. Baroreceptors in the major arteries, like the carotid sinus, sense low pressure. And Ang2, which we know is high when volume is low, also directly stimulates AVP release in the brain. It's another layer of backup.
Okay, so low volume stimulates AVP. What about high volume?
Chronic volume expansion does the opposite. It tends to suppress AVP release, the threshold for release gets shifted higher, and the sensitivity decreases.
Like in the hyperaldosteronism example.
Exactly. Chronic high aldosterone causes sodium and water retention, expanding volume. This suppresses AVP, leading the kidneys to excrete more free water, which can actually cause hypernatremia, high sodium, despite the volume overload.
Are there other triggers for AVP?
Yeah, quite a few. Things like pain, severe nausea, stress, and certain drugs like morphine can stimulate AVP release. Alcohol famously inhibits it, which is part of why it causes dehydration. Clinically, we often see inappropriate AVP secretion after a major surgery or sometimes from tumors producing AVP ectopically, that's SIADH, syndrome of inappropriate ADH.
And thirst. Is that also affected by volume?
Absolutely. Just like AVP, large drops in ECV and blood pressure are extremely potent triggers for thirst, right up there with hyperosmolality. Makes sense, right? If you're losing volume fast, you need to replace it. There's also some link to salt appetite when volume is very low.
Okay, that covers the body's natural systems. Let's talk about intervening clinically with diuretics. What's the basic idea?
Diuretics are drugs that work by blocking sodium reabsorption at different points along the kidney tubule. If you block sodium from being taken back into the body, it stays in the tubule fluid and water stays with it.
So you excrete more salt and water.
Exactly. This leads to a net loss of sodium from the body, a negative sodium balance, which then shrinks the ECF volume. That's why they're useful for treating high blood pressure and edema.
And there are different types.
Lots of different types, classified mainly by where they work in the tubule and their mechanism. You have carbonic anhydrase inhibitors like acetazolamide working proximally, powerful loop diuretics like furosemide in the loop of Henle, thiazides in the distal tubule, and potassium-sparing ones like spironolactone acting on the collecting duct, plus osmotic diuretics like mannitol.
And they don't just affect sodium and water, right?
Definitely not. Because they interfere with various ion transporters, they almost always have predictable effects on other electrolytes like potassium, either wasting it or saving it, calcium, magnesium, and even on acid-base balance. You have to know the side effect profile of the specific diuretic you're using.
How do they get to where they need to work?
Most are organic acids or bases that get actively secreted into the tubule fluid by transporters in the proximal tubule. This concentrates them right at their site of action on the inside surface of the tubule.
You mentioned loop diuretics are the most potent. Why is that?
Well, the loop of Henle, specifically the thick ascending limb where they work, is responsible for reabsorbing a large chunk of the filtered sodium, about 25%. And the segments downstream from the loop just don't have enough capacity to fully compensate if reabsorption is blocked there.
Whereas if you block sodium reabsorption more proximally,
The loop and distal segments can often pick up the slack, reabsorbing more sodium further down, blunting the overall diuretic effect. That's why proximal diuretics are generally weaker.
does using different types together help?
Sometimes, yes. You can get synergistic effects by blocking reabsorption at multiple sites simultaneously.
But long-term use can be tricky. The body fights back.
It does. When diuretics cause volume depletion, all those compensatory mechanisms we talked about kick in, RAS activation, sympathetic drive increases, AVP might go up, ANP goes down. The kidney itself can even adapt over time, with downstream tubule segments sometimes hypertrophying to try and reabsorb more sodium. This can lead to diuretic resistance where the drugs become less effective over time.
Okay, bringing it all together then, this constant juggling act between volume and osmolality. What's the body's ultimate priority when things go really wrong?
The general rule, the one to really remember for clinical situations, is that the body will almost always prioritize defending the effective circulating volume, even if it means letting osmolality slide a bit.
Like in shock or heart failure causing hyponatremia.
Exactly. Maintaining blood pressure and perfusion to the brain and heart is paramount. The body will retain water via AVP to support volume, even if that water dilutes the sodium concentration below normal. It's a life-saving trade-off in the short term.
Is there ever a time when osmolality wins?
Yes, the main exception is severe dehydration, leading to significant hyperosmolality. In that specific scenario, the extreme concentration becomes the dominant threat, particularly to the brain.
So even if volume is also low.
The drive to correct the hyperosmolality takes precedence. AVP and thirst will be maximally stimulated to get water into the system and dilute the body fluids, even if it doesn't fully restore blood volume. It really underscores how sensitive the CNS is to concentration changes.
Wow. It's an incredibly complex but really elegant system when you see how the pieces fit together.
It truly is. The way these feedback loops sense changes and orchestrate responses using hormones, nerves, and the kidney is just remarkable. Understanding is so fundamental.
Absolutely. It seems daunting, but breaking it down like this definitely helps make sense of it.
And that's the goal. It is complex, but absolutely manageable when you tackle it step by step.
Right. So, to leave our listeners with something to chew on, think about a patient who presents with both problems simultaneously. Maybe they were lost in the desert, so they're severely dehydrated and hyperosmolar, but they also fell and have significant bleeding, making them hypovolemic.
A tough scenario.
How would those competing priorities, the drive to conserve water due to hyperosmolality versus the drive to retain volume and water due to hemorrhage play out? How might the body's responses interact, and how would that complicate trying to treat them?
Yeah, that's a great clinical puzzle. It forces you to think about which system might dominate and how interventions for one problem could affect the other. It really highlights why understanding this balance is so key.